Good morning, good afternoon, good evening, everybody. I'm very excited to be here with Alex Bick and Kelly Bolton to talk about genetic risk factors for cancer. We'll also talk a little bit about genetic risk factors for cardiovascular disease. We'll talk quite a bit about clonal hematopoiesis. If you don't know what clonal hematopoiesis is or how to spell it, you'll soon find out. That's something that I'm very excited about. And really, in my opinion, the UK Biobank is really the first data set that has really comprehensively allowed people to study clonal hematopoiesis because the whole exome and whole genome sequencing is available for so many people, and particularly available for so many people over the age of 45. Kelly and Alex are both clinicians as well as researchers. And it's really amazing to me. They're both sort of clinicians as well as computational biology researchers. And that I think is really sort of the maximum cool you can be in science. And Kelly's at WashU, Alex is at Vanderbilt, and they're both very interested in thinking about mutations that predispose people to cancers and other diseases over the long term. That's really very cool. Also, just so you know, Alex sequences meerkat genomes. I like to tell people that it's sort of a fun fact. Anyway, so just to jump in, maybe if I could start with Kelly. Kelly, would you explain to us what is clonal hematopoiesis?
Yeah. So I'll give the explanation that I maybe give to my patients. So like, maybe a simple explanation. So what my spiel is usually I say, as we age, we develop, changes in our bodies, including in our, DNA, that's, our genetic code that is important in determining our health and the function of our body. And these genetic changes occur all over in our skin, in our blood, but sometimes you get a genetic change that occurs in the right gene, in the right cell, and that can lead to that cell developing a advantage over normal cells. And so when that happens in a stem cell, which is a type of cell that's important in regenerating our cells and creating new cells, it can lead to a expansion of a type of cell that shares the same genetic change. And so that's called, when it happens in the stem cell in your bone marrow, and our bone marrow is where we create blood, that's called clonal hematopoiesis.
And so how common is that, sort of among individuals above 45?
Do you want me to take that, or Alex, do you want to? Should I keep going?
Go
Just
ahead.
keep
Go
going.
ahead.
You're doing great.
Keep going. This is a normal part of aging. So if you didn't develop clonal hematopoiesis at a certain point in your life, it would actually be abnormal. Because just by chance with every day of our lives, there's going to be a certain probability that we develop a genetic change in the right cell at the right time. So universally, by the time everyone reaches a certain age, there's enough clonal hematopoiesis that you can detect it using standard sequencing technologies.
So to be clear, clonal hematopoiesis is extremely common, actually sort of normal, if you will. But it's not a cancerous state, but it's sort of there are links between this sort of clonal expansion of certain cells and cancer. Can you talk about that a little bit?
Yeah, so um, you know, fundamentally, cancer is caused by a cell acquiring a stem cell acquiring several mutations that give that cell a competitive advantage and lead the cell to becoming increasingly abnormal. That's like the multi step progression to cancer. But you can detect these mutations, the kind of the first mutation and this possible multi-step progression in most tissues. It happens by chance all the time. And so clonal hematopoiesis, while it is a pre-malignant state, the vast majority of people will never go on to develop hematologic cancer, but we know from both longitudinal studies, but also through mathematical modeling, essentially, of leukemia cells, that probably, like, most people pass through this clonal hematopoiesis stage on their way to developing leukemia at some point in their disease course.
And do the genes that have particular variants matter in terms of who eventually develops cancer and who does not?
Yeah, and Bick, I think you should step up to the plate here. I'm getting a little bored of my own voice.
I think what's particularly cool about clonal hematopoiesis is that it is this pre-malignant state. But because we've sequenced blood in UK Biobank, we can look across hundreds of thousands of people, 500,000 people from the exomes, the genome sequencing data, and find who has a large clone. And because we've assayed this, let's say UK Biobank, instead of doing blood, had actually done cheek swabs, we wouldn't be studying clonal hematopoiesis. We'd be here talking today about the the epithelial of the cheek and pre oral pharyngeal cancers or something like that, I suspect. And so we have this ability to use clonal hematopoiesis as this model system, in the same way that in other organisms or other research questions, you can use mice as a model system, because the same process like Kelly described, at first you have the first mutation, then you have a second mutation, then you have the third mutation, happens in every dividing tissue, happens in pretty much every cancer that comes from a dividing tissue. And we can do it at scale. And because we can do it at scale, we can answer questions like you just posed, Ben, to say, what people, if this is almost a normal part of aging, which people who have this are most likely to get cancer, to get terrible, incurable blood cancers like myeloid leukemia. And in large part because the UK Biobank is so accessible to researchers around the world and has collected so much phenotypic data, we can say that what mutations are important, and we can also say what else about the people might help us understand risk. So there's probably between 50 and 100 genes where mutations in these sort of, Kelly called it the right gene. I like to call it the wrong gene at the wrong time, but maybe that's just a matter of perspective here. Where if you get a mutation in one of these bad genes, it causes this clonal proliferation. Out of those, and I say 50 to 100 because the field's a little bit in flux. There's some disagreement. And also thanks in part to UK Biobank and resources that you've built on DNA Nexus, people are always coming out and writing papers about, we found another 3 genes, which is pretty cool. About 3/4 of people with clonal hematopoiesis have mutations in just one of 3 genes, a gene called DNMT3A, which is a methyltransferase that puts methyl group on DNA, TEP2, which takes that methyl group, converts it to hydroxymethyl as the first step in removing the methyl group, and then ASXL1, which I like to just wave my hands and say it's involved in chromatin remodeling. And I think there's a lot of work to be done to figure out what it actually does beyond hand waving. And those genes cause some modest increased risk of blood cancer, but they tend not to cause very much increased risk. And then there's other genes that are sort of more classic signaling genes, like JAK2, for example, or splicing factors. And those seem to have, instead of a 2-fold increased risk of cancer in developing blood cancer, a 10 to 100-fold increased risk. Groups like Kelly's have put these mutations together, and others like George Vaslu and his group in the UK, and Lachelle Weeks and Ben Ebert's group in Boston, have put a number of these factors together to say we can make a score that's not just what mutation you have, also what is the size of the clone, how much of your blood is being made up of this mutant cell, and other factors like do they have early signs of basically disruptions in blood production. Are there early signs that their blood counts are abnormal? And if, and other factors, how old is the person, for example? And so we now have scores that can say, what is the likelihood of someone who has this, normal-ish process of aging in developing blood cancer.
So I have a follow-on question for Alex, and then really sort of a wider ranging question for both of you. And the question for Alex is, I think in the literature, the link between blood cancers and clonal hematopoiesis or vice versa, right or wrong mutation, whatever, is pretty well established. But I think the link between clonal hematopoiesis and cardiovascular disease, while certainly intriguing and obviously with a potential massive health effect, is a little more tenuous. Can you talk a little bit about the evidence that speaks to the relationship between clonal hematopoiesis and cardiovascular disease?
One of the interesting things when clonal hematopoiesis was first described in larger scale data sets in 2014, through 2017 or so, is that we and others noticed that people who have clonal hematopoiesis were dying, and their rate of death was far in excess of the amount of burden of blood cancer. Fortunately, for the world, blood cancer is a pretty unusual cancer, and people were dying of other things. And so one of the early observations was to try and figure out, why are people dying? And this was work initially done in Sid Jaiswal's 2014 New England Journal paper with Ben Ebert. And then we followed that up to say, we found that people seem to be dying of heart disease. It was one observation. We followed it up with a couple more cohorts, second observation in 2017. And then when UK Biobank started releasing exomes, we and a large number of other people started being able to dig into this observation, at much bigger scale. And what I would say, we've learned is that clonal hematopoiesis is this umbrella term of a driver mutation causing clonality, causing a competitive advantage. And across the 50 to 100 genes, all of those genes might cause a competitive advantage, but not all of those genes cause the same downstream disease phenotypes beyond blood cancer. And even within blood cancer, they actually don't all cause the same kinds of blood cancer. Certain of these mutations cause more of a myeloid blood cancer, like myeloid leukemia. Other of these genes that cause clonality cause more of a lymphoid phenotype, like chronic lymphocytic leukemia, which are two, very, very different diseases even within blood cancer. Beyond this, association with different kinds of cancer, different genes seem to cause different other downstream diseases. So DNMT3A, which is among the most common CHIP genes, seems to cause relatively less cardiovascular disease, for example, whereas driver mutations in TET2 seem to cause more cardiovascular disease. Driver mutations in JAK2, even more so than TET2. And so I think some of the sort of confusion, if you will, in the literature right now is our fault from calling this very heterogeneous condition all one thing. That these are all forms of clonal hematopoiesis, but just like we don't say, oh, the person has a mutation, we say, the person has a mutation in BRCA and that's what causes their breast cancer. I think we're going to get to a point where we're more specific in saying, this TET2 mutation is what causes cardiovascular disease, or this JAK2 mutation. And so when there's criticism that, oh, does clonal hematopoiesis cause cardiovascular disease? I can definitely tell you all clonal hematopoiesis does not cause all diseases. And sort of trying to make that mapping between driver gene and different disease outcomes is something that has been ongoing in the UK Biobank, and I think been really a boon to the community. I think the other opportunity is that we have the ability in the UK Biobank to all be working on the same data set. So in genetics, in years gone by, I found one thing with my collection of 1,000 samples. Kelly might have found something different in her collection of 1,000 samples, and it's hard to know how much of the differences were because my population's in Nashville and her population's in St. Louis. With UK Biobank, because the whole world has access to the same data, has, I think, really advanced methods and really reproducibility and rigor. And, as I'm sure we'll talk about later, every time you do something like make a clonal hematopoiesis call in UK Biobank, you actually return it to the data set so that other researchers can benefit from it.
So yeah, actually, it's kind of amazing because Alex doesn't know what I'm about to ask next, but I think he's pre-pended it perfectly, which is the UK Biobank has announced that it's coming out with a single cell data set in late 2024. And we don't know exactly which tissues are going to be there, but I guess the question I would ask you is, what have you done with single cell data sets so far, and what do you intend to do with single cell data sets in the UK Biobank, particularly thinking about these sort of subtype specific clonal hematopoietic links to disease? And that's a question to both of you, of course.
Yeah, I mean, maybe Alex can talk about it more. I mean, we've done a little bit of work like in this largely unsuccessful work. Like one of the challenges with single cell data is that the technology is maybe, I don't know what you'd say, emerging. And like one of the things that you want to do with, like, fundamentally a question that you want to answer with single cell data in the context of clonal hematopoiesis is to compare within one person, like cells that have the mutation compared to cells that don't. But your ability to detect like low VAF mutations using single cell techniques is challenging. But we have been trying different approaches to do this, and like some of them are successful. It largely depends on what type of mutation like you are focused on, like where the mutation lies in the transcript. Like, so there's challenges to this technique, and so probably good for answering like questions about certain maybe recurrent clonal hematopoiesis mutations, but still some work to go to be able to look at this like across the full spectrum of all mutations.
Yeah, I think to extend on that, my understanding is the first project is being led by Nicole Cerzano and her team at Wellcome, where they've done about 5,000, where the goal was to do about 5,000 or 6,000 blood samples at single cell, and they have done really heroic work over the last 2 years to have blood samples, shipped overnight from collection centers and sort of processed in the middle of the night. And so I think it's going to be a really exciting data set. And one of the reasons why I think it's so exciting is that we'll have scale that we don't have before. So right now, when our group publishes single cell data sets, we have 5 cases and 6 controls. And you can learn a lot from 5 cases who have CHIP because of what Kelly said, because you can look at some of the differences within a person. But to go from 5 cases to 5,000 cases, I think is going to really help us develop better tools for all people to analyze single cell data. And it's also going to teach us some interesting things about what Kelly was mentioning about how cells differentiate. But I completely echo the challenges in terms of from a naive observer perspective, you would say, well, why can't you just tell which cell has which mutation? And that's what Kelly was saying is technically very, very difficult to do. But I think it's, again, going to just be a first model of trying to go from, closer and closer to the tissue of interest.
So that said, Kelly, you were talking about, being able to pick out particular subtypes and so on and so forth. We see emerging biobanks like Our Future Health, for example, being reconsentable. Do you see, sort of a future for reconsenting patients that have really sort of interesting clonal hematopoietic profiles from databases like that?
Yeah, that would be, that's really important. I think, in addition to maybe collecting, like you're getting at Ben viable cells, so that you can do single cell sequencing or do whatever, generate iPSCs. You could a really critical aspect of clonal hematopoiesis that we're really in the beginning of developing and understanding is how like what factors influence certain cells, certain clonal hematopoiesis clones to expand versus remain stable. What are the environmental factors, or other kind of genetic modifiers of that of that process? Because really, what's important about clonal hematopoiesis isn't the presence or absence; it's the trajectory. And probably the trajectory has, more to do with it probably will be more predictive of, leukemia risk, a disease outcome risk than a one shot, look at it at one time period. But we really don't have a lot of opportunities yet to study that in large scales. I think Alex has some really cool, techniques that he's developed in his group to be able to estimate that. But ultimately, like, it will be really cool to be able to, like you're saying, maybe consent people for longitudinal follow-up over time. And
for clinical trials, Kelly
for
is
clinical
the
trials,
PI
yeah.
of two really important clinical trials where one of the challenges is where do you find these patients? And
Yeah.
so being able to go back to biobanks, especially where we know these people have a precancer, and say, let's, invite them to participate in trials will be, really a transformative opportunity. And I haven't heard that UK Biobank has any intention of doing that, but I've heard of other, potential follow-up studies in the UK and around the world that are really excited about that possibility. Obviously, there's a huge gap between signing up for a research study, keeping people sufficiently engaged, and then when you call them up, asking them to, to take another step forward and be part of an interventional study. That's obviously a big hurdle too. But it will be a really great resource for driving precision medicine forward.
So we're almost at the bottom of the hour, so I think we can switch gears here and go a little bit off the rails. And so I think I'd like to ask some sort of computational genomics questions and really future of computational genomics questions. But I'd also like to invite the audience in to ask questions. So there's a Q&A module in Zoom. Please go ahead and start putting in questions. But I'd really like to kick it off by saying, okay, well, so as we know, at a very high level, really, both of you and several other researchers have really leveraged whole exome and whole genome sequencing in the UK Biobank to establish sort of clonal hematopoiesis there and really do a lot of sort of groundbreaking work in clonal hematopoiesis. I'd like to ask you a two-part question. First, a more conservative question, which is, can you talk a little bit about some of the tools, some of the techniques, and really some of the personnel who have worked on that? But then the second thing I would like to ask is, if you were to design a sort of population screening program for clonal hematopoiesis, what would that look like? I mean, what would we need to be able to screen folks for clonal hematopoiesis in the population writ large? Because Alex, I think that's what you were really alluding to when we started talking. When you started talking about clinical trials, the next step there is really thinking about population dynamics. So if I could ask you to speak both about sort of the tools you're currently using, but then also what you'd like to see in the future, I think that would be interesting to the audience.
I'm happy to start and then toss it to Kelly. In terms of analysis tools in our group, and I know in Kelly's group too, we really benefit from the fact that cloud resources are, they're not infinite, but they're really, really big. And so to run pipelines to detect clonal hematopoiesis across 500,000 people, even if it takes 20 minutes a sample, a half an hour a sample, that's years and years of compute time. And thanks in large part to tooling that DNA Nexus has built, instead of doing that one at a time, you can do that on hundreds of samples at a time. And so we've really taken advantage of basically taking tools we've previously put into Dockers, packaging them into applets, which is your runtime. Other platforms, we do this through the WDL or WDL markup language, and then click launch and then see what happens. We really take advantage of the fact that computing is a lot cheaper if you are able to do it on spot instances, which are usually half the price or quarter of the price. And when you do things on spot instances, you accept the risk that your job might be kicked off by somebody else's job that's paying full price. But on average, it'll still be a lot less expensive. And then I think beyond that, a lot of what we do is probably not so different than what many people in the audience do with Jupyter notebooks and R, Python, depending, to try and do the actual genotype, phenotype analyses. You want to talk about tools, Kelly, and then we can go back to Ben's question of how do we do this in everybody?
Yeah, and maybe just to add, because Alex, you summarized it really well. Maybe just to emphasize that you really for clonal hematopoiesis specifically, you really shouldn't be using the germline calls like that have already been deposited for the UK Biobank, because the germline variant callers are not optimized to detect low variants at low allelic fractions, which is what you're looking for in clonal hematopoiesis. So both Alex and I have reanalyzed the UK Biobank data using somatic variant callers, which is just really important. And hopefully our data sets will be available soon for other people to use, like under the UK Biobank return policy. But just to keep in mind, important to not use the germline variant callers for clonal hematopoiesis because it will result in you missing a lot of the mutations.
And then I think, looking more broadly, so I think the return policy is really, really important. So, it's cost our group and Kelly's group, thousands, maybe tens of thousands of dollars to do all of this really expensive compute. Not that each one is expensive, but if you do anything 500,000 times, it becomes expensive. And so, we really hope that, again, just to be very clear to the audience, Kelly and I have returned our clonal hematopoiesis data sets to the UK Biobank, and they're in the process of ingesting them and making them available to everybody, so that all researchers can just use the exact results from a published paper rather than trying to recreate the methods.
Yeah.
So we're grateful for the UK Biobank teams that are working on the ingestion, and we wish that there were more of them so this stuff could happen instantaneously or even be self-published. That would, I think, be the ideal, is within the research analysis platform, the RAP. If you could just click a button and make your notebook public or make your data set public within the community of all researchers who have access to UK Biobank, I think that would really accelerate collaboration. how do we do
So
population screening? Let's talk. Yeah, I'll pause there for a
yeah,
minute.
let's talk about populations, and then we have a lot of questions from the audience. So this is great. Go ahead. Let's talk about how we do population screening for this.
Yeah, I think, one of the questions is, will the entire health system be genome sequenced already? So, there's efforts in the UK to do newborn genome sequencing. Obviously, that won't help you so much for detecting clonal hematopoiesis. But I think there may be an opportunity to leverage data that's already being sort of generated and already being paid for by the health system. So, for example, if you're doing tumor normal sequencing for breast cancer, Kelly and team at MSK, Memorial Sloan Kettering, were able to say, wow, we can reuse all this data we've already paid for. The other approach is to make a really, really cheap assay. And so our group has a DNA sequencing assay that costs somewhere around $8 a sample that can just sequence, the 20 or so genes that make up 95% of clonal hematopoiesis. $8 a sample is actually less than a complete blood count at Vanderbilt, where I think we charge somewhere around $25. And so it's not that hard to imagine that if, as the technology becomes cheap enough, that you could just truly screen everybody when they, come in for their annual wellness visits at 65 and older. And I think, as Kelly was saying, we really need some multi-time point data to guide this. So it's not clear if you screen everybody at 65, do you screen them again at 66, do you screen them at 70, or is it you screen them at 65 and if the clone's, not really there, we call it a day. And those are some of the epidemiology studies that Kelly and I are working on and trying to create that kind of body of evidence.
Yeah, and maybe just to add to that, maybe a little more philosophical, I guess, is this idea of like, should we be screening people for clonal hematopoiesis is another question. Like, yes, we can, and we should for population based in within the context of research, like doing these large studies, that that Alex's technology can be used for is fantastic because it'll allow us to answer really important questions about how do somatic mutations interact with our germline and environmental factors to determine who's going to get cancer, basically. But there's no interventions for clonal hematopoiesis, and we know from some studies that, like, for example, Ben Ebert's group, I can't remember who was the first author of that, Alex, the paper where they there was a study basically where people were approached about, would you want to know about clonal hematopoiesis, and like, how would it make you feel if you were told, that you had clonal hematopoiesis? And I know from my own experience that some people get really stressed out, like, and it's actually bad for some people to hear about it because there's nothing to do, but there's no interventions, there's no management recommendations. And essentially this is the equivalent from, we do sequencing, right, for like hereditary cancer syndromes. And the reason why we do that is because there's established recommendations and modifications for screening that should be done. And we know that it improves mortality. So we do it. We do heightened screening or, other maneuvers to try to reduce cancer risk. But with clonal hematopoiesis, there's nothing to do. And so these are essentially like variants of uncertain significance. Yeah, exactly. Thank you, Alex. So, I think it's probably a little unethical to tell people about clonal hematopoiesis without, just like you do when you do germline genetic testing, you have counseling of the patients before you do the study, so that they have an opportunity to opt out of being told of results that don't have clear management implications, like at the present time, to allow some people to not be told about these results. So I think that's really important, like an maybe just idea to think about. We could talk about it more.
So, I mean, one could make the argument, not that I'm necessarily making it, that this could be tantamount, though, to hereditary cancer screening at some point if it were that common. But to parking lot that for a minute and go to computational genomics, because there's a lot of people online, and we can continue the philosophical discussion at the end. So these questions, I think, dovetail off of something that Alex recently said and that Kelly said earlier. What about low variant allele frequency clonal hematopoiesis associated variants? And what is the influence there in terms of cardiovascular disease? And also, really, what are the basic sequencing depths needed for seeing these clonal hematopoiesis mutations?
I'd say that across most studies, most investigators, and I can't say all anymore because there's just been such an explosion in the field in terms of number of papers. But most people find that the larger the clone, the worse the outcome, meaning that if 10% of the blood is made up of a clone, then pretty much across whether it's cancer, heart disease, kidney disease, whatever other phenotype people are studying, larger clones tend to be worse. And we in UK Biobank have access to sequencing depths that are 40x, 30, 40x. With 30 to 40x sequencing, you can detect usually clones down to about somewhere between 5 and 10%. To detect clones that are 2%, you really need 200, 300x depth. To detect clones that are 0.1%, and Kelly and her group have done this, you need sequencing depths that are 10,000, 100,000x, because you just need more and more reads to be confident that if only one cell out of however many hundreds or thousands, so there's a direct relationship between greater sequencing depth, you can detect smaller clones, but most studies, the smaller clones tend to have little to no health consequences. And again, that's true again across most diseases, across most assays. There's, I think, an active debate about, can I confidently say that they have no health consequences at all? And then that becomes more of a discussion about the mutation itself. So for example, there have been cohort studies out of Denmark that have looked very specifically at JAK2 V617F, which is one of the most common CHIP mutations, very high risk for developing polycythemia vera, other kinds of blood disorders. And in people with, in those studies at least, they say even 1% or 2% clones with V617F matter. And so probably it's sort of an evolving story that will continue to evolve as we generate more data. One thing that my group is trying to do now, we'll see if we're successful at it, is to take all the exome data from UK Biobank, put it together with the whole genome sequencing data from the same person. And so then if you have 30x from one and you have 30x from the other, and now all of a sudden you have 60x. And we should be able to make more confident calls with that kind of data. But invite me back in a year and I'll tell you if it worked or not.
I think there's a lot of people, including Liz Worthy and others, who have done some seminal work on that. I'll send you some papers. That's very cool, though. Kelly, did you have more to say?
Yeah, maybe just that so the natural history of clonal hematopoiesis is, in general, like as a generality, is that the mutations arise at some point in our lives, like sometimes in early childhood or even in utero, and then expand slowly over time. So you can imagine, like, for a younger patient population, it would actually be really critical to sequence at very high depth, because you would expect that most of the mutations you would find, even clinically relevant mutations, later in their lives, you're going to be detecting at very low VAFs when they're young. So we've done some studies on this, looking at childhood cancer survivors, and others. For example, Todd Drewley's group showed using the Nurses Health Study that if you sequence, younger women, that even low VAF mutations predict risk of leukemia later in their lives, like when they, later when they reach middle age or older age. So probably depends also on the age of the population, the clinical relevance of lower VAF mutations.
Very cool. So one more sort of genomic question, and then I want to flip back to sort of a more philosophical question, but highlighted by an audience question. And that is, so somebody was asking about some of these CH mutations that may predispose folks to solid tumors. And the question is, what can RNA-seq data, specifically single-cell RNA-seq data, but I guess bulk theoretically as well, tell us about transition from CH to these solid tumors?
Mm-hmm.
So I think the current model of how CH affects solid tumors, is related to the tumor immune microenvironment, which has become, really a cornerstone of cancer therapy with immunotherapies and others. CHIP mutations that Kelly and I study have a differentiation bias towards myeloid cells. And so to the extent that myeloid cells play an important role in the tumor immune microenvironment, that's where we think, or that's my model at least of how CHIP is playing a role in these solid tumors. And so then, using single cell RNA-seq, we and others have found that patients who have CHIP, have different myeloid cells. They're still macrophages, but they're macrophages that are differentially responsive to inflammatory insults, for example. And I think there's work ongoing here at Vanderbilt and around the world where we're trying to make some of those links more clear. But I think that's sort of where to look and how single cell will help us.
That's really cool. So a last one question, and then actually I'm going to give you the final Jeopardy question in advance. So somebody asked about, and this is a common question that we hear a lot. When you're running MUTEC-2, particularly for clonal hematopoiesis or things like MUTEC-2, can you run that in tumor-only mode for clonal hematopoiesis, or do you need tumor normal? But I think the bigger question there is, what general tools do you think folks who are interested in these kinds of things really need to be able to leverage, whether they're using return data from UK Biobank or whether they're starting to look at new data sets and et cetera. So that's, I think, the sort of normal question. And then the final Jeopardy question, if I may just seed this in the back of your head, is what algorithms are we missing in the field? What would you, if you could wave a magic wand and something would pop up at ISMB next year, what would that algorithm look like? But first, let's go to sort of running tumor normal versus tumor only, and then also, is that sufficient to pick up CH? And also, really sort of what tools do labs need to really start working on this kind of stuff?
So I can start off. You can run. So we do run MUTEC-2 as our as a variant caller. We like to use a consensus for things like the UK Biobank where the depth is low and you're actually really only able to detect variants that are above 2% VAF or higher. We like to use a consensus approach because MUTEC-2 is the best variant, I think. Well, arguably probably the best variant caller, somatic variant caller. It's fantastic. But it likes to call some weird stuff, especially indels, like weird indels. So we like using MUTEC-2 plus something else, and we require a consensus calling. And then, but maybe more important than like thinking about running it in tumor versus like matched tumor normal mode is that after you do your somatic variant calling, you have to do a lot of additional filtering. for clonal hematopoiesis because, first you have to distinguish clonal hematopoiesis from germline variants. And second, you have to distinguish clonal hematopoiesis from artifact. So both Alex and I have like similar approaches. Like, we have additional filters basically that we run after variant calling to try to filter that stuff out.
Yeah, I would just completely agree that it's tough, and we wrote a paper describing one approach. Again, it's not like there's only one approach, and I agree with Kelly. The caller is not necessarily the most important thing. Probably the more important thing is trying to do that downstream filtering of what's germline, what's somatic, what's artifact. We published a paper on this led by Caitlin Blashart, which I think Brent can pass on. And then we have a long-suffering manuscript with Jessica Regan, who's at Duke, who I think is on the call today, where we're trying to make this into a protocols paper. So you could just say step 1, step 2, step 3. And I promised myself for the past 2 years that it was going to be, imminently we would be done with this paper, but hopefully by announcing on the webinar, we'll actually get this across the finish line sometime in the new year.
Awesome. So final Jeopardy question. If you could wave a magic wand and have a new algorithm or pipeline, what would that be?
Yeah, I think tools to make this curation process better, faster, easier is really important. And so can we think of making something like Nomad, where it's just a public data set. There's no access restriction whatsoever. That just says across all of these studies, this mutation, JAK2 V617F, we see all the time. So if you see it, it's probably real. And so I think trying to both make a reference data set and then make a better algorithm to try and query one of these reference data sets to update the prior probability, if you will, that a mutation is real, would be really helpful. We've tried various kinds of machine learning on this, and at the end of the day, we still haven't been able to beat the manual curation of an expert hematopathologist. But it's a new year with large language models, so maybe we'll get there. So over to Kelly.
Yeah, I agree. And one other thing, so we for example, spend a lot of time because we're interested in clonal evolution studies, trying to develop techniques specifically for calling very, very low VAF variants, like less than 2%. And there's really not a lot of great resources for variant calling specifically for that context. And one other thing that's really lacking are gold standard data sets. So, we had to create our own gold standard by taking AML tumor samples and diluting them to validate how well we were doing and try to improve things. But really, if there were more publicly available reference data sets like this, that would really help benchmark the tools that we develop and to compare tools against each other.
Bringing it back to the UK Biobank, if you had a small fraction of patients where we know they had CHIP, and again, if in some hypothetical future recontact was allowed, just being able to do a cheek swab and be able to do the genome sequence on the cheek swab, genome sequence on the blood, and be able to compare the two, and that would really accelerate methods development to try and differentiate between what's real and what's not.
That is extremely interesting. There's nothing sort of set in stone, but would longitudinal proteomics analysis help in that case?
I don't think so. But maybe it would if you wanted to, if we had proteomics data for DNMT3A and you wanted to validate that a loss of function, I guess DNMT3A mutation, resulted, I guess, in lack of, but really more than
if they went out and collected this sample necessary to do the proteomics, they also So it would help in some sense, but not from the data itself. This is
Yeah.
one of the frustrating things about biobanks. Everyone just assumes the DNA is the same, and it's not. And we've shown this clonal hematopoiesis. We've shown it with telomere, length changes. Shown it with the mitochondria changing. And sort of the list of things that change in your genome gets longer every year. And so, really, what we need is actually a second whole genome sequence time point from some subset of UK Biobank.
Yeah.
We're trying to do that at small scale in the Framingham Heart Study, but obviously having the full richness of all the other omics that are available and phenotype data in UKB would make it really, really special.
Yeah.
Wow. That is certainly a tall order, but it is not impossible that WGS will get inexpensive enough to do a double whole genome sequencing in large biobanks at some point. But I want to thank the panelists very much, really for a very entertaining discussion, and thank the attendees for lots of lively questions. This will be recorded. The slides will be available. Maybe. But it will definitely be recorded. You can check it out and have a great day. Take care, everybody. Thank you.
Thanks, Ben. Thanks, Alex.
Thanks for having us.
Yeah.